Thank you for the opportunity to present our work here. So since 2022, I have co-led a lab with Jim and Sarah at the WeHi, and we focus on brain cancer. But today I want to present our foray into spatial metabolomics, which is a new technology for us. So I first want to convince you that metabolites are really what you should care about. So they are the intermediate or ultimate endpoint of all life-sustaining chemical reactions that happen in a cell. So hence measuring their abundance, unlike measuring any of the components of the upstream pathways that makes them, will directly inform you about what the cell is currently doing, right? So like measuring bulk RNA sequencing, measuring bulk metabolite abundance is actually quite cheap, quite comparable to RNA sequencing. But the difference really emerges when we consider wanting to do it on a single cell level or even a spatial level, where spatial metabolomics is roughly a third to a fifth of the cost of single cell or spatial. So one stunning example of the power of what spatial metabolomics can do is this recent study by Capolupo. And they looked at lipid abundance at subcellular resolution, and they could show that there are some lipids that are really involved in determining the intracellular heterogeneity versus some that are involved in determining the intercellular heterogeneity. But before we really dive into the computational strategies for that type of data, I have to tell you how we can actually measure metabolites in space. Here I'll focus on MALDI-TOF, which is probably currently the most popular technique. So what you do for MALDI-TOF is you have your frozen section, you mount it onto a glass slide, and then you embed it into a matrix. So importantly, the matrix is just a material that will allow the desorption and ionization of the metabolites on the sample surface. Then you are going to ablate that sample surface of the embedded tissue using a laser in a grid-like pattern, and everywhere the laser hits, metabolites will be released, and they can then be measured by a time-of-flight analyzer, where the velocity of how fast the metabolite travels is actually inversely related to its mass, giving us a spectra. And this then, obviously, over the grid-like pattern, will allow us to visualize any detected mass-to-charge ratio in space. All right, so this technology produces an incredible amount of data. We're talking 10 to 100 gigabytes per slide, and requires really a computational pipeline that marries both aspects from traditional mass spectrometry and spatial transcriptomic analysis. So the first hurdle with this type of data is that you're going to have to convert the proprietary file formats from the instruments into something that is open source and then it provides interoperability. So we use R and the Cardinal pipeline for any of our processing of our data downstream. And this will allow us to identify the peaks and then annotate metabolites later on. However, because of the scale of the data, we actually have to do this detection or the peak detection in field of views in order to reduce the computational burden. This does not impede our ability to visualize any then detected metabolites across the spatial range. Beyond this, we have also made some improvements that will allow us to really make this pipeline more robust, in particular in the peak identification and in the metabolite annotation. So one of these is something that we have done to the standard workflow. So typically the standard workflow just includes normalization, spectral smoothing, baseline correction, peak picking and alignment, and peak binning, all from just the tissue from the matrix with the embedded tissue. But we also do that entire pipeline from just the matrix without any tissue embedded. And this allows us to determine any peaks that are coming just from the matrix alone and allows us to remove these, giving more accurate data. After peak identification and metabolite annotation, the next step is going to be visualization and downstream analysis, where we rely on the R ecosystem. So for example, we can use kernel density estimations from the stats package in combination with the plotly package to really visualize these 3D plots of metabolite abundance in space. And then using a really new package called RampDB, we can look at pathways, for example, or even investigates regions where we have metabolites, differences in metabolite abundance. Unfortunately, after you've sorted all of this out, this is not the only hurdle that you have for MALDI-MSI data. With this technology, the matrix the sample is embedded into drawing data is entirely going to determine what metabolites you can detect. And here this is illustrated by the ability to find cholesterol in 6 different sample preparations for brain. So we set up an experiment that compared 5 different sample preparations for both polarities. You can do this in negative polarity or in positive polarity, again, giving you quite different metabolites. And because we were interested in brain cancer, we used a xenografted CT2A tumor. And we did all of this in collaboration with Metabolomics Australia, and then developed a pipeline to evaluate the sensitivity and the specificity. So sensitivity is typically the only thing that is evaluated when optimizing sample preparation, and consists really of just asking how many metabolites can I detect? And this is in terms of unique peaks and annotated metabolites. But we extended this sensitivity analysis by also investigating what biological pathways are represented. And in our specific example here, we found that both analyses pointed really to the recrystallized CHCH in positive polarity as a standout performer. But to evaluate specificity of the different sample preparations, which again is not typically assessed, we ran 2 further experiments. And so for this, we used the sequential slides of the same xenografted CT2A mouse tumor, where we dissected the normal from the tumor and then ran these samples separately using RNA sequencing and LC-MS. So for the comparison with LC-MS, what we found was that over 40% of the metabolites detected by LC-MS were also found by this recrystallized CHCA matrix in positive polarity. And then furthermore, we could show that half of the metabolites also shared the same direction of change between tumor and normal compared to the LC-MS, and this was really the highest consistency. We repeated this type of consistency analysis when comparing RNA sequencing to our MALDI-MSI results, but this time we used pathways because that is the common unit that actually connects those 2 different modalities. And again, we could show that recrystallized CHCA in positive polarity had the highest consistency, with 66% of the pathways having concordant change of direction. So what is really cool was that the discovery of this matrix and optimizing this really had instantaneous sort of results for GBM research. So Justin Lathia at the Cleveland Clinical Center had observed that spermidine was significantly upregulated in brains of mice with tumors compared to those that only had sham treatment. And spermidine also happened to be one of our most interesting results, as it showed a significant difference between the tumor and the normal. So by collaborating with us, Justin could show that this increase in spermidine was indeed localized to the tumor region and did not just happen ubiquitously in the brain of mice with tumor. While our analysis regarding spermidine used some of the aspects of the spatial data, it could have been done by just dissecting, right? So next we asked whether we could use the data a little bit more cleverly by identifying niches. So for this, we applied a convolutional neural network technique that was previously published. And this approach uses the raw peak abundance data and then uses UMAP to reduce that data to 3 dimensions. And this can then be visualized in space as RGB values. And this provides the input to a convolutional neural network that allows the segmentation of this type of data. And what was really nice to see was that our convolutional network is very good at identifying the tumor. But beyond this, it's also really interesting that there are niches that are forming at the tumor border. And these show elevated abundance in signaling pathways and lipid metabolism, which we're now following up. And the way we are following this up is by combining spatial metabolomics with spatial transcriptomics, as well as histological information. So to this end, we obtain serial sections of the tumor, which we use alternatingly to generate H&E, spatial transcriptomics, and spatial metabolomics. And in the future, this will allow us to identify the cell type composition and histological features that are associated with these particular increases in metabolic activity. So, but this it just remains for me to thank the lab who are doing all of the hard work, in particular Ty, who was an honors student in the lab, as well as Sarah and Jim, who obviously co-lead the lab. Thank you so much.
Thank you very much. Any questions for Saskia?
Thanks. We're doing something very similar actually in South Australia with this. Can you comment on what proportion of your peaks are you able to identify with HMDB? And I guess now when you have that multi-omics, there's probably a lot you can do with those kind of unidentified peaks potentially, right?
Yeah, I actually don't know. We never really looked into sort of how many of the peaks we can't identify. That hasn't been our biggest problem. Our biggest problem has been that we actually, for the peaks that we can annotate to the HMDB, we often annotate 2 to 10 metabolites to that particular peak. And that then leaves you with asking the question, which one is it actually? And this is why the spatial transcriptomics data is going to be so handy, because it's hopefully going to be able to point to what metabolite was actually there. So yeah, we typically identify thousands to like 3,000 of metabolites. So I've never been super concerned about, like the not identified peaks, but more concerned about, can I actually annotate and do I actually know what I'm seeing?
We managed at one point to actually, I think we put kind of like gene identifiers almost on our peaks and plugged it into like an RNA-seq pathway analysis. And it worked surprisingly well, actually, at least in our context, for like identifying a bunch of different immune things. Have you tried any pathway type stuff?
Yeah, so that's what I was showing. There was pathway. So yeah, pathway stuff works really brilliantly. And as I said, this RAMDB package is really a game changer for that type of analysis.